AI and data science star. Good afternoon. Thank you very much. It's an honor to be here presenting in the NJ AI and Data Science Stars seminar series. So today I'm going to present a real-world example of the importance of expert data science, fair sharing of information, in a talk that's entitled Protein Data Bank from Two Epidemics to the Global Pandemic to mRNA Vaccines and Paxlovid. So my plan is to tell you about the critical role that structural biologists, data scientists, and the Protein Data Bank, the PDB, I'll call it, together played in fighting the COVID-19 pandemic. Well, the punchline of my talk was put succinctly by Dr. Anthony Fauci, who was the former head of the National Institute of Allergy and Infectious Diseases. In a New York Times magazine in 2023, he cited the importance of 3D biostructure information and said, show me a person who's vaccinated, got infected, took Paxlovid, and died. I can't find anybody. So I'm now going to explain why Fauci made these statements and put the global efforts of structural biologists, data scientists, and the Protein Data Bank into context. So my talk's going to cover the history of the Protein Data Bank and the Worldwide Protein Data Bank Partnership, the RCSB Protein Data Bank, which is headquartered at Rutgers University, which is the US data center for the Worldwide Protein Data Bank Partnership, our one-stop shop for 3D biostructure information, rcsb.org. And then I'll spend the bulk of my time explaining how structural biologists, data scientists, and the PDB played decisive roles in fighting against SARS-CoV-2 and taming the COVID-19 pandemic. And I'm going to close with a sobering postscript that will, I think, convince you that this was all completely preventable. Completely preventable. So the PDB was established as the first open access digital data resource in all of biology. It was founded on the 20th of October, 1971. We celebrated our 54th birthday a couple of weeks ago, with just 7 X-ray crystal structures of proteins, and it's been continuously funded by the United States government ever since. On the right panel, you can see 4 of the Burley structures that motivated protein crystallographers, as structural biologists were known in those days, to establish the PDB. These are illustrated in the right panel. They include, this is also a pointer, right?
Yeah, but then you
okay.
speak.
Yeah. So they include myoglobin. That's the protein in muscle that stores oxygen, that makes it available when you want to move. It's also what makes meat red. So when you see a nice steak in the supermarket that's got that nice red color, that's coming from the myoglobin. And when the myoglobin gets oxidized, it changes color to brownish. So that's why the old meat in the supermarket looks kind of brownish, and you shouldn't buy it. You should only buy the meat that looks red with the fresh myoglobin. Hemoglobin, that's what makes your blood red. That's the oxygen carriage molecule and some of the very first enzymes for which structures were determined. Enzymes are, for this audience, and I'm sorry if I'm talking down to you, I don't mean to. Enzymes are proteins, machines, molecular machines that catalyze chemical reactions inside cells. And life is only possible with these proteins to speed up the chemical reactions that are necessary for life. So the archive has grown more than 30,000-fold since that time in 1971 to become the single global resource for experimentally determined atomic-level 3D biostructure information. At present, the PDB houses nearly more than 245,000 structures of proteins, nucleic acids, meaning DNA, RNA, viruses, and macromolecular machines. This information is important because in biology, 3D shape dictates function, either biochemical function or biological function. So knowledge at the atomic level of 3D shapes tells you about function, tells you how biology works. That's why it's so important. That's why the federal government has been funding the Protein Data Bank for more than 54 years. recognizing the global importance of the Protein Data Bank. It's actually been managed jointly since 2003 by a global partnership, the Worldwide Protein Data Bank, which includes data centers in the US, meaning Rutgers, Europe, Japan, and most recently the People's Republic of China, plus 2 specialist data resources that house the experimental data from electron microscopy and NMR spectroscopy. These are alternative tools for determining 3D structures at the atomic level. So reflecting our very long-standing commitment to the FAIR principles, so that's findability, accessibility, interoperability, and reusability, and the FACT principles standing for fairness, accuracy, confidentiality, and transparency. These are emblematic of of responsible data stewardship in the modern era. Our commitment means that PDB data have been distributed under the most permissive terms using the Creative Commons CC0 license, which means that the information can be downloaded anytime by anybody in the world at no charge and with no limitations on the use of this information. We were previously accredited by the Core Trust Seal, and in 2022, the Global Biodata Coalition designated the PDB as a global core biodata resource, and I quote, of fundamental importance to the wider biological and life science community and the long-term preservation of biological data. The RCSB Protein Data Bank that I lead serves as the US data center for the WWPDB. We're supported jointly by NSF, NIH, and the Department of Energy. We, at Rutgers, and with 2 smaller sites at UC San Diego, at the San Diego Supercomputer Center, and UC San Francisco, operate 5 interlocking services that together convert global data into global knowledge for the public good. We support research and education across fundamental biology, biomedicine, the energy sciences, and biotechnology, serving literally many millions of PDB data depositors and PDB data consumers, working in nearly every sovereign country and territory recognized by the United Nations. The only place we don't download data to are countries that are in the middle of a civil war in Africa, basically. Even North Korea downloads our data. Among non-biological researchers, meaning you, PDB data are used by chemists, computer scientists. How many are in the room would call themselves a computer scientist? Got to be a few of you, right? Okay. Mathematicians? Any mathematicians in the audience? Data scientists in the audience? A few more hands, okay. Physicists, statisticians. And finally, PDB data are used intensively by all of the major biopharmaceutical companies and many of the smaller biotech companies. Again, they download the data for free, they use it, no limitations on what they can do with it. So once the data are archived in the PDB, once they pass through our data intake system at Rutgers, our research-focused web portal, rcsb.org, serves as a one-stop shop for open access to more than 245,000 experimentally determined PDB structures, alongside more than 1 million computed structure models coming from AlphaFold DB and the Model Archive. So how many of you have heard of Google DeepMind? Everyone in the audience should put their hand up. Everybody, yeah. Google DeepMind created AlphaFold, which ultimately generated the AlphaFold database. Could not have done that without the Protein Data Bank, without open access to the PDB. In our world, we designate experimental structures with an Erlenmeyer flask and computed structure models, which are not as accurate on average, with this icon of a display screen. We also make the quality, the reliability of this information very clear and make a distinction between the experimental structures and the computed or predicted structures. So this slide shows the homepage of rcsb.org. It's probably the most important slide of my talk. And highlighted within the red ellipse is a toggle that allows you to opt in or opt out of access to more than 1 million complete structure models, or CSMs. So when you move the toggle from the left to right, you can study both the universe of all known protein structures and more than 4 times that number of predicted structures that fill in the gaps for a variety of different organisms. All of this can be done with advanced search, and I'll show you the advanced search page on the next slide. This is probably the second most important slide of my talk. And can you see the? Oh yeah, it's barely visible. There's a whole series of different search terms that one can use either with or without computed structure models to find the structures or the computed structure models that you're interested in. It supports full Boolean logic. And all of the functionality on this advanced search page is actually available through APIs. In fact, the entire website is powered by underlying APIs. These APIs can be accessed directly, enabling programmatic and searching and bulk data download. So I want to now move on to the story of COVID-19 and viruses. So over the years, PDB data have proved to be essential for responding to emerging viruses. Since the early 2000s, the world has faced down 3 major outbreaks of coronavirus infections that jumped the species barrier from some animal to human. SARS coronavirus, which was first detected in 2002, infected ultimately about 8,500 individuals worldwide, killing just over 800 people between 2002 and 2004. I'm going to refer from now on to SARS-CoV, just to SARS-CoV-1, because at the time, of course, we didn't know that there was going to be a 2. But so I'll call it SARS-CoV-1 to avoid confusion. The next coronavirus epidemic that hit, caused by the Middle East respiratory or MERS coronavirus, struck in 2012. And thus far, MERS has killed about 900 individuals, and it remains a public health threat in the Middle East and parts of Asia, where it's actually endemic in camel populations. So despite these 2 very clear warning signs, few countries prepared for the possibility of a much more serious coronavirus epidemic and the possibility of a pandemic. So fortunately for us, with the benefit of hindsight, we now know that fortunately for us, structural biologists and data scientists in the Protein Data Bank laid the groundwork for an ultimately successful response to the inevitable 3rd wave of coronaviruses. Effective mRNA vaccines were designed and antiviral agents were discovered and developed with the benefit of open access to PDB structures of SARS-CoV-1, MERS-CoV, and SARS-CoV-2 proteins. SARS-CoV-2, the unseen enemy, is shown here on the right in an artist's rendition of a respiratory droplet in a painting that came from our former artist in residence, David Goodsell, who authored the RCSB PDB Molecule of the Month articles for 25 years. He ended up publishing 300 articles, one every month, for 25 years, and explained biology and explained the importance of 3D structures to many people over the time through our educational website, pdb101.rcsb.org. That's a way of getting acquainted with the data. So the organization of the SARS-CoV-2 genome is depicted schematically in this slide. So all coronavirus genomes are very long, single-stranded, positive sense, 5' capped, 3' polyadenylated messenger RNAs that are ready for translation by host cell ribosomes. As soon as the viral RNA gets into a human cell, our machinery takes over and starts making the coronavirus proteins. Most of the non-structural viral proteins are actually expressed within a pair of polyproteins, where the protein chains are actually all strung together in a line. And 2 enzymes, 2 viral proteins, are required to actually process these polyproteins to make the infection move forward. One of them is a papain-like proteinase, that cuts at 3 sites in the dark blue here. And the other is the more important is, and the star of this show is the main protease, which cuts at all these other sites, chopping up the polyprotein into individual protein chains. Without the polyprotein, the processing, without the functional main protease, the infection cannot proceed. So the very first structure of a SARS-CoV-1 protein was that of the main protease, rightly. It has a protein data bank identifier, 1 Quebec 2 whiskey, using the NATO alphabet. And I say it was the right choice, because if the viral polyprotein cannot be cleaved to release all the individual proteins, the infection cannot proceed. This structure was determined in 2003 by my company, SGX Pharmaceuticals, where I led R&D as the chief scientific officer, as Mengjia told you during her very kind introduction. It has the distinction of being the only PDB structure with its primary publication appearing in the New York Times. We didn't actually publish a paper. We simply made the data available, and we told a reporter at the New York Times that we'd done this, and he featured our work in one of the articles in the Science Times. We did this because we were an oncology-focused drug discovery company. We had no interest in infectious disease, but we did have the technology to quickly make this information available to anybody who wanted to actually build build a drug against SARS-CoV-1. So as soon as we put the data into the protein data bank, we released it to the public so that anybody could work on viral countermeasures. And thus far, you can see that more than 125 structures of the SARS-CoV-1 main protease have actually been put into the protein data bank by researchers from around the world. That was one Achilles heel of the virus. The second Achilles heel, as I mentioned before, is the papain-like proteinase that cuts at only 3 sites. Without the action of that enzyme, the infection can't proceed. The first of these SARS-CoV-1 PL-Pro structures contributed to the PDB was made public in 2006, so that's after the epidemic actually dissipated. And to date, more than 10 structures of this second SARS-CoV-1 protease have been contributed to the archive. The first PDB structure of the entire extracellular portion of the SARS-CoV-1 spike protein was determined using single particle cryo-electron microscopy and only released to the public in 2017. That was still 3 years before the pandemic hit. And to date, more than 70 SARS-CoV-1 spike protein structures have been contributed to the PDB. These structures told us about the pre-fusion versus the post-fusion conformations, the 2 different functional states of the protein. It showed us how the spike protein is recognized by the cellular receptor, which is angiotensin converting enzyme 2, a protein that sits on the surface of our cells. And also looking at the way various antibodies recognized and neutralized the spike protein. In 2013, we saw the first MERS-CoV protein structure come into the PDB. And to date, more than 70 spike protein structures have been contributed to the archive, 40 M-Pro structures and 10 PL-Pro structures. And we come back to SARS-CoV-1 and MERS-CoV protein structures in a minute, because they were crucial for actually guiding design of the SARS-CoV-2 vaccine when the pandemic hit in 2000. So when the pandemic hit, once the sequence of the virus was released, my colleagues doing structural biology around the world sprang into action and responded to the pandemic and began putting their data into the PDB. So together with our partners at the Global Worldwide Protein Data Bank, we prioritized getting those data in, rigorously validating and then biocurating them, and getting the data out to the public. And we urged our depositors to the data into the public domain as soon as possible and not wait till the The structures were published, and to the credit of most of our contributors, they did just that. They simply made the data available, which is not something that people would have done historically in a different context. We created a coronavirus resource page on our web portal. We update that every week with each new release of the data archive. Every week, we are putting somewhere between 300 and 400 new structures out into the public domain. These are coming to us from all over the world, and we're distributing those. We also update our education-focused portal, the pdb101.rcsb.org, and this hand-washing video that you see down here that explains why it was important to wash your hands during the pandemic has been viewed more than 500,000 times. We authored various articles for the public and had a lot of traffic to the website as people were thirsty for information, hungry for information about the virus. So the right-hand panel of this slide summarizes what is now our nearly complete atomic level 3D knowledge of SARS-CoV-2 protein structures, and they're embodied in more than 5,500 structures of the protein data bank, that have been made public since early 2020. That's more than 2% of the entire archive has been devoted to understanding SARS-CoV-2. The left-hand panel of this slide shows SARS-CoV-2 attached to the surface of a human cell and just disgorging its messenger RNA into the cell to begin the infection. And it's this entry process that is blocked by the vaccine. So let me just digress for a moment and tell you a little bit about my journey. As Manjir described, I did 13 years at Rockefeller University as a Hughes investigator, became a chaired professor, became a chief academic officer of the university. I thought they were going to bury me at Rockefeller. I was convinced I was going to do that. That was my future. I would simply spend my entire career there. Fantastic place to work on the Upper East Side of Manhattan. Housing provided at a price for the postdocs and graduate students. And in 2001, 2002, I began to realize that I felt I had stopped learning. I wasn't learning new things. I was just doing the same thing every day. And I realized I needed a change. So I decided the thing to do was to go to do drug discovery, structure-guided drug discovery in industry, and I moved to SGX Pharmaceuticals, took that company public in 2006 on the NASDAQ, sold it to Lilly in 2008, and then part of the deal was I had to spend 4 years at Lilly as a distinguished Lilly research scholar. And at the end of it, I got a payout, and then I retired. I had no plan, and unfortunately, or maybe not unfortunately, but not surprisingly anyway, I failed retirement completely. I realized I didn't want to stop working. It was just too interesting. So I was very lucky in that Rutgers came along and picked up the pieces. And so in January 2013, I joined the university and took over the RCSB protein data bank. I decided at that time that I was not going to continue doing experimental science, which is what I'd done my entire career. I was going to reinvent myself as a data scientist and focus my research on the information that was stored in the PDB, and we've been doing that ever since. And in the last 5 years, we put in a lot of effort in trying to understand COVID using the data in the archive. So the first thing we did during the first 6 months, about 6 months into the pandemic, was actually try and understand how the viral proteins were evolving as a function of time. So during the summer of 2020, when everything was shut down, we could only work with students virtually. We had a team of 11 undergraduates and one very talented high school student who's now a senior at Columbia in engineering, They worked with 6 faculty members online to analyze how each one of the viral proteins had changed in amino acid sequence and changed in 3D structure using genome sequence information that we were downloading from the GISAID web resource. So they had sequences of all these different isolates over 6 months from all over the world. And you could see, when you looked at those isolates, how the disease had been spread by aircraft. People who were sick got on planes, went to another country, transmitted the virus there. So we ended up analyzing nearly 50,000 translated viral genome sequences that were available in June of 2020. And then we ultimately published a paper in 2022 that reported how these proteins underwent change, how they evolved during the first 6 months of the pandemic. And what we learned was that actually there was very little change. And the reason for that is that all coronaviruses have an enzyme that corrects errors in replicating the genome. It's a proofreading exonuclease. It's known as non-structural protein number 14. Coronaviruses have to have that because their genomes are so long. Without that, there will be lots and lots of errors in terms of the genome replication, and the viruses could easily end up with a non-functional protein, and then be unable to go on and infect a cell. So in nearly 90% of the cases, we found that the sequences of the viral proteins didn't change during the first 6 months of the pandemic, which is really quite amazing. And when changes did occur, they were largely just one amino acid at a time, meaning that one base had changed in the messenger RNA. And we found that most of the observed substitutions actually mapped to the surface of the protein, where they were predicted to have little or no effect on the structural stability of the protein. But of course, for the spike proteins, what that allowed for was immune invasion. So changing the surface of the spike protein allowed the virus to evade whatever antibodies the host had developed as a consequence of either a vaccination or a prior infection. We also found that the hydrophobic cores of the viral proteins, which are essential for their 3D structural integrity, were almost unchanged. And we found that the active sites of the enzymes that the virus depends on for its lifestyle were also unchanged. So what that told us was these viruses were good targets for drug discovery because they're not changing during the course of the infection. So you might wonder, you might wonder now, and you might have wondered in the past, why it was that SARS-CoV-2 went on to cause a global pandemic, whereas SARS-1 and MERS just disappeared from the population. So there are 2 reasons. The first is that SARS-CoV-2 is capable of what's called pre-symptomatic or asymptomatic spread. You can be sick, you can be infected with the virus, but not feel it, not know it, and just walk around and infect people. That's what was happening on these aircraft. The most successful viruses are actually those that in the universal viruses are those that can be transmitted during the incubation period. So they just keep going, keep going, keep going. The victims of SARS-CoV-1 and MERS-CoV could only spread the infection when they were acutely ill, meaning that when they were hospitalized. So that meant the virus didn't spread, those viruses didn't spread very much. They infected healthcare workers, and they were disproportionately numbered among the casualties and the fatalities from SARS-1 and from MERS. But it didn't affect the wider population, and that's why the viruses died out. We isolated the infected patients in hospitals, supported them. Some of them died, some of them did not, and we were able to kill off the infection that way. So the second reason that SARS-1 and MERS didn't become pandemics was because they had a much higher case fatality rate than did SARS-CoV-2. A successful virus doesn't kill the host because that's going to limit spread. So the successful viruses just make you sick, and then you spew virus over everybody around you. They get infected, and it's not going to work if you die. So the viruses that are more successful are the ones that don't kill their infected hosts. In fact, hang around long enough to be passed on. Remember, it's all about getting that that piece of genomic information to the next generation and the next generation and the next generation. That's true whether you're dealing with viruses or people. So the very first PDB structure of the SARS-CoV-2 spike protein, so that's ID 6 Victor Sierra Bravo, was released to the public in February of 2020. That's just weeks after the sequence information for the virus became publicly available, literally only weeks. Today, there are more than 2,000 SARS-CoV-2 spike proteins publicly available in the PDB. And like the earlier spike protein structures that I told you about, they explain the viral lifestyle. They explain the importance of the spike protein and how the virus works. The very first mRNA vaccine designs that were generated in January of 2020 were not based on structural knowledge of SARS-CoV-2 spike. They were based on structural knowledge of SARS-CoV-1 spike and MERS spike. These are very, very similar proteins in terms of their amino acid sequence, and even more similar in terms of their 3D structure. So when designing the inaugural Moderna vaccine, this is mRNA-1273. How many had a Moderna shot during your time? How many had a Pfizer shot during your time? More, yeah. I've recently only had been getting Pfizer shots. So what Moderna reported in their paper was that when they designed their spike protein vaccine, they used information in the PDB that showed them that if you have a double proline modification of the protein, so that's a change in the RNA sequence leading to a change in the protein sequence, that it stabilizes the spike protein in a highly immunogenic fusion conformation, in a pre-fusion conformation. And I believe the same thing was true for the vaccine designers at BioNTech, which became the Pfizer vaccine, because their inaugural vaccine also has this double proline change in it. And in fact, every mRNA COVID vaccine that's been made since 2020 has that double proline change. And the key with that double proline change is it makes the protein that gets expressed by the cells that take up the vaccine, it makes it much more immunogenic. The vaccines that relied on the wild type sequence didn't do nearly as well. Those vaccine designers didn't read the literature and they didn't look at the PDB. So the rest of the story is well known to you. Two highly effective, nearly identical vaccines, mRNA vaccines against COVID-19 were brought to the market by Operation Warp Speed, literally in record time. And to date, these vaccines have been, and their successors have been administered to more than 5.5 billion people worldwide with impressive results. So the vaccines that were designed with the benefit of open access to PDB data, accredited with saving tens of millions of lives globally and preventing severe forms of infection in hundreds of millions of people. So you've got to love the protein data bank. I mean, that is staggering. So I'm going to turn now from vaccine design to the discovery of small molecule anti-SARS-CoV-2 drugs. So very early in the pandemic, drug repurposing was the order of the day. Companies that had drugs on the market tested them against SARS-CoV-2 to see if they would work. And there are two such examples, Gilead's remdesivir and Merck's molnupiravir, both hit the RNA-dependent RNA polymerase, which is actually a three-protein, contains three distinct proteins, two copies of one of them, the dark blue, one copy of the light blue, which is where the enzymatic activity resides, and one copy of the light blue. The two copies of the dark blue are like molecular chopsticks holding the messenger RNA of the virus and the copy of the RNA that's being generated by the RNA-dependent RNA polymerase. And if you kill this enzyme, you kill the infection. So Gilead discovered that remdesivir works. That's an IV drug, so that's fine in hospital for an acutely ill patient, but that's not really very suitable for use as an outpatient basis. Merck discovered that their molnupiravir, which is a pill drug, a pill form drug, an orally bioavailable drug. Merck discovered that worked against SARS-CoV-2. So both of these got an approval from the US FDA, an emergency use authorization. The Merck's molnupiravir was actually the product of work at Emory, targeting Venezuelan equine encephalitis virus. So no reason that it should have worked, but it did. That Venezuelan equine encephalitis virus has been seen here in the Northeast over the last few years, interestingly. So both of these structures that I just showed you explain how those drugs work at the atomic level. So they were a short-term solution, but certainly not a long-term solution. So what I'm going to tell you now is how structure-guided drug discovery was used to discover a drug that would hit the SARS-CoV-2 main protease, the principal Achilles heel of the virus that I described earlier. It's the target of Pfizer's highly effective, orally bioavailable small molecule drug known as Paxlovid. That's a fixed-dose combination of an active ingredient called nirmatrelvir and another drug known as ritonavir, and I'll explain ritonavir in a moment. So the PDB currently houses nearly 1,800 crystal structures of SARS-CoV-2 Mpro, the first of which was deposited in January, just literally a couple of weeks after the gene sequence of the virus became available. So within 2 weeks, a team in Shanghai synthesized the gene, expressed the protein, since it made synthesized a DNA gene that expressed the protein, purified it, crystallized it, determined the 3D structure, and put it in the protein data bank. When we did that same work 20 years earlier with SARS-CoV-1, it took us about a month. The technologies got a little bit better, but not actually a lot better in the intervening years. So they made this information publicly available by putting the structure into the PDB. But I want to turn back the clock now and tell you what the nirmatrelvir origin story, and that actually begins in 2000 with the discovery and development at Pfizer, interestingly, of a compound that they called PF00835231. All drug companies have these very long generic names for the small molecules that they synthesize. And I'm showing here in the right panel how it works, blocking the action of the SARS-CoV-1 main protease. So Pfizer intended that this drug was going to be used as an injectable for in-hospital antiviral use in acutely ill SARS-CoV-1 patients. It was the product of a structure-guided drug discovery campaign that began in 2003 that was facilitated by open access to my structure of SARS-CoV-1 main protease. Now, unfortunately, but totally understandably, Pfizer stopped the program, the drug discovery program, in the mid-2000s before going on to clinical trials because SARS-CoV-1 had disappeared and the company correctly judged that there would be no commercial market for an anti-SARS-CoV-1 drug at the time. Situation changes later, of course. In early 2020, when the pandemic hit, Pfizer reactivated the SARS-CoV-1 project with the goal of discovering and developing an orally bioavailable antiviral agent for outpatient use their discovery was facilitated by open access to the very first structure of the SARS-CoV-2 main protease that I described earlier. So the result of their efforts was nirmatrelvir, the active ingredient of Paxlovid, and shown here in this right panel, inhibiting the SARS-CoV-2 main protease. So you've got to love the PDB. They went from basic science information to a drug using the information that was in the PDB. So both nirmatrelvir and the earlier compound that was active against SARS-1 that I showed you on the previous slide are covalently acting inhibitors of the enzyme. The way they work is that they're recognized by the active site of the enzyme. The enzyme catalyzes a single turnover reaction, a single chemical reaction, which results in a blocked active site because the drug is covalently attached to the protein, and that shuts down the virus, shuts down the main protease, shuts down the viral infection. Nirmatrelvir as a small molecule does have one major drawback, and that is that it's rapidly metabolized by one of the liver enzymes, cytochrome P450, CYP3A4 it's called. So our livers, located right here, are chock full of enzymes that detoxify stuff that we eat. Millions of years, billions of years of evolution led to this so that early man could survive eating stuff they just found in the middle of nowhere. And even though it might have been somewhat toxic, the liver would detoxify. What that means is when you ingest a drug, sometimes the system, this defense system in the liver, neutralizes the drug. So the solution that Pfizer settled on was to co-administer nirmatrelvir with ritonavir. So this is a potent CYP3A4 inhibitor. It prolongs the half-life of nirmatrelvir by preventing its degradation as it passes through the liver. So Paxlovid, this fixed-dose combination of the two drugs, received emergency use authorization from US FDA in 2021, less than 2 years after public release of the viral genome sequence. And the speed with which Pfizer was able to move was unprecedented. It often takes more than a decade to go from knowing the target to getting a drug and getting it on the market. At the end of my talk, I'm going to come back to the fact that nirmatrelvir also works against the main proteases of SARS-1 and SARS-2 and MERS, in addition to SARS-CoV-2. And that's what's going to lead me to my sobering postscript and the claim that all of this was preventable. So a little bit about ritonavir. The PDB houses more than 120 structures of human cytochrome P450 CYP3A4. And the right panel of this slide shows how ritonavir blocks the action of this enzyme, gums up the active site of the enzyme, and prevents it from degrading nirmatrelvir. This drug was originally developed by Abbott, now called AbbVie, as an HIV protease inhibitor. It explains in 3D why ritonavir has been useful for prolonging the half-lives of other viral protease inhibitors that are susceptible to CYP3A4. So the hepatitis C drugs, for example, are co-administered with ritonavir to prevent their degradation. So this was an established way of developing antiviral agents for safe use in humans. So this is not the fact. So returning to the fact that nirmatrelvir inhibits the main proteases of SARS-2, SARS-1, and MERS is not actually surprising. In 2021, with some summer undergraduates now able to be in person after the easing of the pandemic, they worked with me and Professor Sagar Kare, a member of the Institute for Quantitative Biomedicine that I founded. Our goal was, working with these students, was to understand how the amino acid sequence, 3D structure, the active site, and the peptide substrate cut site diversity across all known coronaviruses. We wanted to understand why it was that this drug works on 3 different coronaviruses. The work's not published. The grad student went off to work for an AI company, has been very busy ever since, so we haven't quite got it written up yet, but I'm going to summarize the data for you. So we know that all coronavirus main proteases fall into one of 4 genera, which is shown here in this cladogram, and this is based on clustering of the full-length amino acid sequence of the enzymes. SARS-1, MERS-CoV, they show up in the same genus, the beta coronavirus genus, shown here in light blue. And I just ask you to just see how close they are to one another in this diagram, and remember that. The image shown in this right panel explains how the active site of the enzyme can be partitioned into 6 pockets. And these 6 pockets bind different amino acids in the peptide that gets cut. The site of inactivation by the drug, by those 2 drugs that I told you about, is right here between P1 prime and P1. So that's where the cut actually occurs. So if we consider only the amino acids that line the active site of the enzyme and do another round of clustering, what we can see is that MERS, SARS-1, SARS-2 are even closer together when you consider the active sites. So the active sites of the enzyme are more conserved than the rest of the protein. That's not surprising. We see this all the time with enzymes. The business end of the molecule that's responsible for catalyzing the chemical reaction generally doesn't change during the course of evolution. That stays fixed. Other parts of the protein can change. So this very busy diagram, I don't expect you to read it with any comprehension, just looks at the conservation of the amino acid residues that are lying in the active site for all known coronaviruses, and it's really quite amazing. So each column on the right shows the frequency of each amino acid at each position in the active site. And what you can, dark blue means it doesn't change. Dark blue means it's the same. And what you can see is that there are certain places in the active site where no change occurs. That leucine, that proline, that phenylalanine, that cysteine, there's just no change whatsoever across all of the coronaviruses. Their enzyme active sites for the main protease essentially remain the same throughout the evolution of the virus, the coronavirus family. 12 of the positions of the 12 of the amino acids that line the active site are essentially invariant across evolution. So again, based on the structural information, we can also understand the conservation of the peptide sequences that are recognized in the polyprotein when the enzyme goes in to process the polyprotein to cut it up in all the pieces. And what you can see in the following slide, again using a similar convention, is that at this position, there's only one time that it changes. Only one time. And essentially only around 1% of the time. 99% of the time, that position is always a Q. And if we look at P1 prime, that was, let me just go back, sorry. Okay. What we're doing is we're looking at how the identities of these amino acids change as a function of evolution. This one is essentially invariant. This one, there are only 3 options. This one, there's also a restricted number of options. So P2, P1, P1 prime, a lot of blue. What that tells us is that the cut sites can't evolve either. They have to remain the same. So the virus active site remains the same. The cut sites remain the same. That's why Paxlovid works, nirmatrelvir works against SARS-1, MERS, and CoV-2, because not only did the enzyme active site not change, the cut sites also didn't change. There's double selection pressure on the virus to preserve those key functional elements of the proteome. So we use the information that we've gleaned in this analysis of all coronaviruses to make some predictions as to which other coronaviruses nirmatrelvir will work for, and we're going to publish that soon. It's encouraging. it won't work against every coronavirus. We don't believe that, and we've actually managed to show that in some cases it won't work against certain coronaviruses, but it will work against many coronaviruses. So we may have a drug for SARS-3. So this brings me to the sobering postscript. This is my last data slide. Now that we understand the constraints on the evolution of the coronavirus main protease active sites and the cut sites, what we learn is that there's been a failure in the free market for emergency pharmaceuticals. So my late father was an economics professor, and he told me on more than one occasion that a failure of the free market is defined as an inefficient distribution of goods and services within the economy. He believed that repetition was the key to learning. I think that's true. Market failures occur when the incentives for the individual or the individual company for rational economic behavior don't lead to a rational outcome for everybody. So what I mean by that is we cannot always rely on capitalism to get the right answer when it comes to emergency pharmaceuticals. The government has to step in sometimes. So with the benefit of hindsight, we now know that investment literally of 200 or 300 million US dollars in continuing the SARS-1 drug discovery program at Pfizer would have given us the possibility of saving millions of lives worldwide before the Moderna and biotech mRNA vaccines came online. And it would have prevented prevented the shutdown of economies, and it would have prevented the global economic losses to the tune of trillions of US dollars. So you can understand why after SARS-1, everyone said, okay, it's over, thank God. Only 800 people died, no problem. Let's just move on. But when MERS hit, it was no longer, I think it was apparent to the experts that it was no longer a matter of if, simply a matter of when the next coronavirus epidemic would strike with the attendant risk of becoming a global pandemic. So experts knew this. So thanks to structural biologists and the PDB, experts also knew at the time that the active sites of SARS-CoV-1 and MERS-CoV were almost identical to one another. The SARS-CoV-1 and SARS-CoV-2 active sites are 100% identical. The amino acids that stick into the active site, that bind the substrate and cut the polyprotein, they're identical in the two enzymes, identical. There's one difference between SARS-1 and MERS enzyme active site, one difference. They knew that these sequences and structures were highly conserved. So unfortunately, they ignored that information. What I think they should have done was to recognize, God, this could become a real problem for us if it becomes a pandemic. But what biology is telling us is, what evolution is telling us, is that if we find the right drug company, Pfizer in this case, and fund them, we could have a drug for the next one. That never happened. But fortunately, structural biologists studied SARS-1, SARS and CoV, MERS-CoV proteins, and they put their data into the open access PDB, and that laid the groundwork for engineering of the life-saving vaccines that I told you about, and for the discovery and development of a highly effective antiviral. So Pfizer made a huge investment initially in SARS-1, and that set the stage for very rapid discovery and development, and ultimately emergency use authorization, and ultimately full use authorization of Paxlovid. NIAID, Moderna, Pfizer also invested very heavily in developing mRNA vaccine technologies. And for me, as an oncologist, one of the few silver linings of the COVID-19 pandemic, other than Zoom works now, is that our experience with SARS-CoV-2 revolutionized how vaccines are designed and manufactured, how they're tested in the clinic, and how they undergo regulatory approval. So over the next few years, we're going to see new mRNA vaccines emerge for cancer. And independent of what's happening with the federal government, with mRNA vaccines and vaccines for childhood illnesses, all the major companies are moving ahead with programs like this because they see that there's a possibility of making vaccines that actually effectively treat cancer. I'm very, very optimistic that this is going to be provide benefit. I'm also hoping that we can get beyond the current misguided approach to vaccination and think more clearly about pandemic preparedness worldwide, because all of this could have been prevented if Pfizer had been given $250 million of US taxpayer money. That's all it would have taken. Conservatively, that's all it would have taken. Probably would have taken less to show that they had a safe drug that could be administered to humans. That could have been used in 2020 to save people's lives around the world and prevent having to shut down whole economies and prevent all those economic losses. We didn't do as good a job as we should as scientists in communicating that information to the federal government. And I urge you, as you think about your roles going forward, to try and become the best scientific communicator you can, because without trust of the public, there will be no money at NSF, there'll be no money at NIH, DOD, NASA, all the places that you are looking to get money from. And it could have a catastrophic impact on the US economy. Other countries are not doing this. Other countries are moving ahead full speed, understanding how important science is. And I'm referring specifically to PRC. More patents are coming out of PRC than anywhere else. Top universities in the world, 9 out of 10 of them are now in the PRC. They're not in the West. Only Harvard managed to stay in the top 10, and it isn't number one. It isn't ranked number one. It's actually, I think, maybe number 10 or number 9. I don't remember the exact ranking that it got most recently, but it's, it's not encouraging. So to recap, I have explained how open access to research data generated with public and private money, particularly thousands of atomic level 3D structures of coronavirus proteins that are archived in the protein data bank, enabled basic and applied researchers to combat the COVID-19 pandemic and literally save tens of millions of lives. So I would ask you to join me and Dr. Fauci in loving the protein data bank. It's a terrific place to go to do research. You can think of it as a matrix which has 245,000 rows and tens of thousands of columns of data that's highly curated, expertly validated, underpinned by a data model. I haven't mentioned this in my talk, underpinned by a data model that's very robust and was good enough that Google DeepMind could use the PDB data directly, train AlphaFold, and produce AlphaFold 2 and revolutionize protein structure prediction. There's a lot more to be gained by mining and studying what's in the PDB. So I thank you for listening. I know I've gone over time a bit, and I apologize, but we did start a bit late. I need to thank the 60,000 or so structural biologists worldwide who put their data into the PDB, who made all this possible. I also thank the many millions of basic and applied researchers and their trainees, their students around the world who utilize PDB data, because it's their enthusiasm for what we do that ensures that we get funded and ensures that the data resource is going to live for another 50 years. We're in our 55th year of operations now, and there's every reason to believe that 3D structure information is going to remain important in biomedical and biological research for as long as it takes. I thank the NSF, NIH, and DOE. I thank Rutgers, UCSD, and UCSF, and our Worldwide Protein Data Bank partners. And this huge team of people here is what keeps the train running. It's a combination of structural biologists, PhDs, most of them, data scientists, software developers, IT professionals. All of those tools have to come in. And we're now using developing AI tools to actually make us more efficient, to make what we provide to you, the users, more useful. And we're going to continue for as long as we can delivering these data and these services to millions of people worldwide at no charge and with no limitations on usage for the benefit of everyone. Thank you very much.
Thank you, Stephen. So any questions?
So some high-level questions. So what do you think of the potential impact of AI to PDB in the next 5 or 10 years? So in your opinion?
So the holy grail right now, is to understand two problems. One is how to be able to predict effectively, accurately, how small molecules bind to target proteins in the computer, as opposed to having to do. Each one of those structures costs, on average, $100,000. The replacement value of the PDB is 24 billion in planning. So we need ways of predicting how the small molecules bind to the target proteins, because that's going to accelerate drug discovery. If we combine that utilization of the AI technology with the results of machine learning and all the data that are in the drug companies that explain the connection between a chemical that's put into a person and side effects. So side effects from small molecule drugs are typically the result of binding to other proteins other than the target. So this is called off-target toxicity. We do not understand the determinants. We do not understand well the determinants of how small molecules bind to unwanted proteins, the off-targets in the human proteome. That's the source of the side effects. We need to minimize that while maximizing the binding to the target protein. So we can, I think from AI and machine learning, we can learn an awful lot. Sorry for the pun. We can appreciate an awful lot about the connection between the structure of the small molecule and whether it's likely to cause a side effect and what side effect it's likely to cause. A bit of nausea, if you're going to die of cancer, may not be a bad thing, right? But a bit of nausea in a drug you have to take every day of your life is not a good outcome for the average patient. What it's going to lead to is, I feel like taking a drug today, it doesn't make me feel very good. I think I could give a drug holiday. People do this all the time. People on anti-HIV medications take drug holidays, allowing the virus to suddenly come back to life in their bodies, because they don't never eradicate the virus from the host. It's simply, they can block its replication with antiviral drugs. You take a drug holiday and boom, the virus, the virus goes wild. So that's one problem that we need to solve. Both how we bind to the desired target and often understanding to avoid binding to the unwanted off target. Second thing we need to understand, we need to be able to predict with much better fidelity, is how, when two proteins come together, they can bind together tightly, how they can recognize one another. So this is particularly important in the immune system. How is it that the antibodies recognize the spike protein? If we learn more about how proteins interact with peptides, how proteins interact with other proteins, we will do a better job with discovering and developing drugs that modulate the action of the immune system. Two ways we want to be able to do this. One is to tamp down the overactive immune system in inflammatory diseases like rheumatoid arthritis. And there are antibodies on the market that do quite well right now, but they still have a lot of toxicity associated with them. That's typically on-target toxicity. So we need to make better antibodies, better monoclonal antibodies that with longer half-lives, easier dosing and all that, lower cost of goods for treating an immune system that's proactive. We also need better drugs to make the immune system more active against cancer. So the checkpoint therapies. So you know the story of Jimmy Carter, right? You saw his funeral. Most of you have seen something about his funeral in the last 12 months, maybe a little. I don't remember exactly when he died, but it was about 12 months ago. About 15 years ago, Jimmy Carter had stage 4 widely metastatic melanoma, and he was at death's door. He goes to the church in Plains, Georgia, where he had been baptized as a child and where he'd worshipped his entire life. So that his family home was in this little community on a peanut farm. It all sounds very, but it's sort of true, actually. He goes to the church and he says, I've had a wonderful life, but I want you to know that I'm very soon going to die of metastatic melanoma. And I want to thank you for everything you've done for me and for my family. But I'm here to say goodbye. I'm probably going to go into hospice in a matter of weeks, and I'm going to pass away probably this year. Everyone's very sad. Great man, tremendous humanitarian, successful post-presidency ever, arguably. Not so successful as a president, but very successful after he finished the presidency. 3 months later, he comes back, he's looking a lot better, and says, you know what? I don't think I'm going to die. The doctors, the oncologists at Emory in Atlanta, at the medical school there, blood from Merck, it's called Keytruda. And I'm one of the lucky 1/3 of individuals with melanoma. When they receive that drug, they either get a long-term remission, possibly even a cure. We now know that Jimmy Carter was cured by that drug. It's a monoclonal antibody that prevents the cancer from downregulating the immune response to the cancer. It upregulates the immune response to the cancer. We need better drugs to do that. That will come from AI, I'm very confident, using AI tools combined with the data that are in the PDB. Fantastic. It's absolutely fantastic.
So the PDB group will also develop your own AI algorithm, or you mainly generating or collecting or providing data?
I don't understand your question, sorry.
So yeah, you mentioned that using AI maybe help you for those two tasks you mentioned, right? The prediction of sample binding or something like that. So the PDB group, your group member will also develop an AI algorithm, or
We are developing AI tools to serve our users better.
Okay.
We're not doing drug discovery.
You don't do
We're not funded to do drug discovery.
or like a prediction algorithm. Also
So we are working with the OpenFold team at Columbia. I don't know where we are in the building right now, but Mohammed Al-Karishi is leading a very successful team at Columbia. OpenFold and the OpenBind, which is trying to get better at predicting hospital molecules bind to protein targets.
yeah. Okay, so you still focus on data generation and
We're focusing on delivering this information in the most effective way to our customers. Because we're funded to the tune of north of 10 million a year by NSF, NIH, and DOE combined to make this available, this information available in the world. And we want to see more users, more power use of the PDB as opposed to one-at-a-time use, more the use of the APIs to harvest the information, build training data sets, develop new insights into biology, develop new insights into protein physics, when you name it. I mean, there's all sorts of possibilities. Someone at the back wanted to ask a question. Yes, sir.
Basically, this might be a vague question, but you said the COVID-19 could have been could be prevented if Pfizer had enough support from the government.
So we could not have prevented SARS-CoV-2. We could have done a much better job of managing the pandemic, and we could have managed it in a way that didn't. If we had had a drug from Pfizer, which didn't involve shutting down the world economy.
My main curiosity is that where are we at now right now? Because is there any virus out there that we should be worried about, and are we actually prepared for the next?
So I should have added one more slide. I apologize. So the most obvious potential pandemic is avian influenza, bird flu. So the good news is we know that every avian influenza protein looks like at the atomic level. We already have the equivalent of Paxlovid available to us for treating influenza virus infections. One is an inhaled agent, not very usable in a broad setting. The other is a pill. So in the short term, we will probably be able to. So when an outbreak in a rationally operating public health system, when an outbreak, and I don't think we have one right now, but in a rationally operating public health system, when an outbreak of bird flu were to happen somewhere in the Midwest, where there are these huge chicken farms, where they're producing chicken meat and eggs on an industrial scale. If it were to emerge in an area like that, the health officials in the state would swoop in. They would establish what's called a cordon sanitaire around the area. They would go in and they would vaccinate everybody they could inside the cordon sanitaire with an anti-flu vaccine that was tailored, mRNA vaccine that was tailored to the particular strain of influenza virus, avian influenza virus that in that community. And they would make drug broadly available for free to anybody who got infected. That would be likely enough to contain it. But what we know, if you start using that drug actively, resistant strains of the virus are going to emerge and you need new drugs. The data are there in the PDB to support structure-guided drug discovery of the next generation of anti-influenza agents. So, with the right in the event of a catastrophe, we could be okay. What we don't know right now is whether or not our Department of Health and Human Services would actually respond in the right way. There's no guarantee whether the CDC, et cetera, would actually respond in the right way. We don't know. So we put to the test. I pray we're not put to the test because we might find that the response is hampered by ideology and not science, and adherence to ideology and not to science. I'd like to think at the end of the day, it's going to prevail and the right things are going to be done. But I don't think we can be as confident of that as we might have been in 2016. Yes, sir.
One of the main problems that you said about this data bank is to the bindings between the molecules itself. So, like you have the Rosetta synergy for that, right? Already. So why do it MLP instead of in one unit?
Sorry, you got to speak up. My hearing is not what it used to be.
So we have the Rosetta's energy to study the bindings between the molecule itself. I believe it's a physics-based comparison. Why do you instead of that?
Oh, well, so, okay, so you raise a really interesting point. Have you heard of Harold Shiraga?
No, sorry.
Okay, so there were a lot of papers published over decades. Harold Shiraga was a very prominent chemist at Cornell University, Ithaca, New York. Very famous, won lots of prizes, never won the Nobel, but won lots of prizes. Every year he would publish a paper and say, next year, my physics-based methods for predicting how proteins fold, how proteins recognize one another, how small molecules bind to protein will be perfected and the problem will be solved. Every year he published a paper that essentially said the same thing. And every year he was wrong. Until the approach that David Baker first pioneered, which was to use evolutionary covariance to try to understand long-range contacts within folded proteins. So what Baker realized, so he got the other half of the Nobel Prize in chemistry last year for protein design. What he realized was the amount of DNA sequencing we've been doing. There are situations where we have hundreds of predicted amino acid sequences from genome sequences, in which we most of the time see that an amino acid at this position is big, and an amino acid at this position is always big and this is always small, except very occasionally this one becomes small and this one becomes big. That tells you that they touch one another in 3D in folded protein. So he used that evolutionary covariance method to improve those data with success. What they did was to combine that evolutionary covariance approach with multiple sequence amendments with the contents of the PDB and machine learning and produce AlphaFold 2. So physics-based methods alone don't work, haven't worked. And so the question is, why not? Because you would think, okay, well, physics is, I'm a recovering physicist. That was my original training, mathematical physics. My deviation into biology was totally unexpected at the beginning of my academic career, but that's undergraduate career, that's fine. The reason physics-based methods don't work is because we're not very good at working with entropy, and we're not very good at working with. So the free energy of stabilization of a protein is about -10 to -20 kcals per mole. Proteins are balanced on the edge of stability, and there are probably good reasons for that in terms of protein turnover inside cells, chopping up the protein and then reusing the amino acids to make other proteins because the needs of the cell have changed. If you had a protein that was super stable and once you made it and it never got degraded, that would be a problem for a cell because you couldn't recover the amino acids. So if you take a protein and you in aqueous solution and you transfer it to 100% organic solvent like benzene, for example, and when you study that protein in the crystalline state, what you will learn is that the water molecules that decorate the surface of the protein are still there. Occasionally, there will be a hydrophobic patch on the surface of the protein where a benzene might be found. Interesting observation, to be sure. On the surface, but and then in the second shell, you begin to see fewer. You might still see a few water molecules, and then there's benzene around it completely. Otherwise, it's benzene. So what that tells you is that there's an integral part of the protein structure. And this all has to do with the evolution of the entropy changes that occur when a protein folds. So in an extended polypeptide chain, you have certain amino acids that are hydrophobic. Ordered collections of water molecules called platelets surround those hydrophobic amino acids. But when those hydrophobic amino acids are brought together, when they come in from the hydrophobic core, those very low entropy water molecules get ejected into bulk solvent. And the entropy of the system goes up. And that's what it minus T delta S increase, delta S is positive, minus T delta S then means the protein, ensures that the protein is going to be stabilized, folded state. So we're really not very good at leveling that. We're not very good at understanding the hydrophobic effect, which is what I just talked about. That's why the physics-based methods don't work. Because if you think about it, if a amino acid side chain is making a hydrogen bond with water, it's just making a hydrogen bond with another amino acid in the polypeptide chain, the energetics of that, the enthalpy of that is the same. The system doesn't care if the hydrogen bond is removed from itself. It's the entropy changes that matter when the protein folds. So that's, I think, why the physics-based methods don't work. Now, I think the future is in the combination of physics-based methods and machine learning and fold in AlphaFold, etc. That's where I think the future lies. And getting much better at handling water. And with that, I think I need to take a break.
No question, right? So, yeah, let's thank Dr. Burley again. Yeah, thank you for the excellent thoughts. Our data science students work on this data set.
Thank you again. It was an honor. Thank you.
Thank you.
And your introduction, only my mother would have believed. Thank you.